Hello, as you know, I think, beta, we are discussing about the functions of plasma membrane. You know that plasma membrane is selectively permeable. It allows only the useful molecules that enter the cell, and it allows that only that the molecules like secretion and waste they leave the cell. So for it enables the cells to maintain the homeostasis. That is a constant internal environment in spite of changes outside it. So the function of this plasma membrane, it permits the entry and exit of only some molecules, and it is that's why we call it a selectively permeable membrane. And studies on the function of plasma membrane, they have shown that it performs certain physical activities such as diffusion and osmosis for the intake of such substances, and during biological or physiological activities such as active transport and endocytosis are performed by the plasma membrane. Today I'll discuss about the diffusion. What is the diffusion? Suppose when any substance like any molecule ion or any molecule like CO2, water, they can move across the plasma membrane through a process called diffusion. The substances are of very small size. They can diffuse easily through the phospholipid bilayer of the plasma membrane. So if you see, like if you open the perfume bottle at one corner of your room, it will spread. The perfume smell will spread all over the room. So how it is possible? Because it is due to the diffusion. Diffusion is a spontaneous movement of molecule from the region of high concentration to the lower concentration. जहाँ पे आपने परफ्यूम की बोतल ऑन की है वहां पे कंसंट्रेशन ज्यादा है और ये ज्यादा से लोअर की तरफ मूव करता है और इट इज ए स्पॉन्टेनियस यू डोंट नीड एनी एनर्जी until uniform concentration is finally achieved. So diffusion is the fastest in the gaseous phase than in the liquid and solid. Okay. So there is an activity, it is given in your book, like how you can check whether it's a diffusion. The other is osmosis. Water follows the law of diffusion also. The osmosis is a special type of diffusion. Here the spontaneous movement of water molecule through a selective permeable membrane, suppose plasma membrane, is called osmosis. In the osmosis, mainly the movement of water across the plasma membrane of a cell is affected by the amount of substance dissolved in water. Osmosis is a passage of water from a region of high concentration through a semi-permeable membrane to a region of low water concentration. जैसे अगर आपने जैसे आपके घर में आपने देखा होगा कि मदर दाल वगैरह या चने वगैरह भिगो देती हैं, तो वो जो वाटर की कंसंट्रेशन है जो भिगोते हैं उसमें क्या वाटर का कंसंट्रेशन इट इज हाई आउटसाइड द seed or that pulse, so it will move from outside to the inside. Okay, so osmosis is a purely a mechanical diffusion process by which cells absorb water without spending any amount of energy because it is from high concentration to low concentration. So suppose if you put a animal cell, suppose RBC or plant cell, into a solution of sugar or salt that is prepared in water, so there are three possibilities. If the medium of the cell that surrounds the cell has a higher water concentration than the cell, okay, means the solution is very dilute because cell की भी कोई concentration है. अगर आपने बाहर कोई एक ऐसा मीडियम सेल को दे दिया जिसमें बहुत dilute solution है, the cell will gain water क्योंकि अब सेल के अंदर the cell will gain water by osmosis. Okay, such a dilute solution is called hypotonic solution. एक वर्ड आता है बेटा हाइपोटोनिक, एक होता है हाइपरटोनिक. हाइपो का मतलब है जहां पे बहुत डाइल्यूट सॉल्यूशन है, जो सेल की कंसंट्रेशन है उससे भी डाइल्यूट है, तो वाटर कंसंट्रेशन बाहर ज्यादा है, तो दैट्स व्हाई इट विल एंटर्स इनटू द सेल. ओके, तो सेल विल गेन वाटर बाय ऑस्मोसिस. Such a dilute solution is called hypotonic solution. And while water molecules are free to pass across the plasma membrane in both directions, the more water will enter the cell and then leave. The net overall result is that water enters the cell. In such a situation, cell is likely to swell up, like become inflated or turgid. Such swollen RBC may ultimately burst or hemolyzed if the outside water is totally like diluted. तभी हम जैसे ब्लड को या किसी भी चीज को जब भी हम लेते हैं तो एक पर्टिकुलर कंसंट्रेशन होती है हमारे पे, एक बफर होता है जिसमें हम रखते हैं ब्लड को. अगर हम सिंपल वाटर के अंदर सेल्स डाल देंगे लाइक आरबीसी या डब्ल्यूबीसी तो इट विल ब्रस्ट, क्योंकि बाहर वाटर की कंसंट्रेशन ज्यादा है, सेल के अंदर वाटर कंसंट्रेशन कम है, तो water will move inside the cell, so that will burst the cell. Okay, that is due to the osmosis. So there is definitely 2-3 questions from this point of view, either diffusion or osmosis in your question paper. The other is isotonic solution. Isotonic का मतलब same, जो बाहर है वो अंदर है. If the medium surround the cell is of exactly the same water concentration as the cell, then there will be no net movement of the water across the plasma membrane. Such a solution is called isotonic solution. अगर जो सेल के अंदर वाटर की कंसंट्रेशन है और वही बाहर हो जाए तो नेट मूवमेंट वाटर का नहीं होगा. तो देयर इज एग्जांपल लाइक रिंगर सॉल्यूशन इज एन आइसोटोनिक सॉल्यूशन फॉर द एनिमल सेल. हर सेल्स का अपना अलग अलग आइसोटोनिक सॉल्यूशंस होगा. जैसे हम कोई भी एक सॉल्यूशन बनाए और जो उसमें सेल की कंसंट्रेशन वाटर की कंसंट्रेशन है वही अगर बाहर हो तो नेट मूवमेंट वाटर का क्या होगा, जीरो. So in this case, the water crosses the plasma membrane in both directions, but the amount going in and the amount the going out is same. So there is no overall movement of water. Such a situation, the cell will maintain the same size. In the third situation, if you put the cell into the hypertonic solution, hypertonic मतलब जैसे आपने पानी के अंदर बहुत ज्यादा चीनी घोल दिया, नमक घोल दिया, तो उसमें क्या होता है, if the medium has a lower concentration of water than the cell, it is very concentrated solution, the cell will lose water by osmosis. Okay, अंदर सेल की वाटर की कंसंट्रेशन ज्यादा हो जाएगी, बाहर कम है, तो वाटर अंदर से बाहर की तरफ आएगा. Cell will lose water by osmosis. Such a concentrated solution is called hypertonic solution. हम देखो प्रैक्टिकल लाइफ में इसको यूज करते हैं. हम जो अचार डालते हैं, मुरब्बा डालते हैं, उसमें क्या होता है, either we use a pickle में हम high concentration of oil, high concentration of sugar या high concentration of salt या हम यूज करते हैं, मतलब we are using hypertonic solution, जिसकी वजह से बैक्टीरिया वगैरह अगर उसमें कोई ग्रो करे भी तो नहीं कर सकता, because the cell will life. ठीक है, due to this plasmolized. In this case, like water cross the plasma membrane in both directions, but this time more water leaves the cell than enters. Therefore, the cell will shrink, and in this situation, the plant cell is said to be plasmolized, and animal cells को हम बोलेंगे is said to be crinated. Plants के लिए word use किया plasmolized, और animal cell के लिए हमने word use किया RBCs के लिए crinated. Okay, beta. And definitely 2-3 questions will come from this angle, diffusion or osmosis regarding hypotonic, isotonic or hypertonic. Now, what is the difference between diffusion and osmosis? I will explain. So the experiment, that is osmosis with an egg and osmosis with raisins and apricot, definitely one or two question will come from this activity. So listen carefully. The activity 2.4, 2.5 and 2.6. Definitely there will be question from these activities. Okay. So please prepare yourself. Diffusion can occur in any medium, but osmosis occur only in the liquid medium. The diffusion molecule may be solid, liquid or gas, but in the osmosis it involves movement of solvent molecules only. Semi-permeable membrane is not required in diffusion, but semi-permeable membrane is required in case of osmosis, and diffusion is dependent upon the free energy of the molecule. And diffusion in osmosis, it is the diffusion of solvent molecule, so it is influenced by the presence of other substances also. And you can also say that osmosis is a special type of diffusion. Now the second one, there's a difference between uh, like types of mediated transport. One is called facilitated transport, and second is called active transport. You can say like diffusion or active transport. जो diffusion है, diffusion is a slow process. Like हमने गैस एक्सचेंज में बात की, जैसे हम सांस ले रहे हैं, ब्रीदिंग में गैसेस एक्सचेंज हो रही हैं ब्लड में, बॉडी में, या ब्लड एब्जॉर्ब कर रहा है न्यूट्रिएंट्स को, दैट इज थ्रू द डिफ्यूजन. हमारी बॉडी में ज्यादातर प्रोसेस जो है वो विदाउट एनी एनर्जी यूज हो रहे हैं. इट इज ए स्पॉन्टेनियस प्रोसेस एंड डिफ्यूजन इज गोइंग ऑन हायर टू लोअर कंसंट्रेशन. डिफ्यूजन हमेशा होगा हायर टू लोअर, तो इट इज ए स्लो प्रोसेस. और जो एक्टिव ट्रांसपोर्ट होती है, कभी हमें अगेंस्ट द कंसंट्रेशन भी काम करना पड़ता है, अगेंस्ट द कंसंट्रेशन ग्रेडियंट, दैट इज ए रैपिड प्रोसेस ऑल्सो. डिफ्यूजन के बारे में पहले बता रहा हूं मैं. इट इज स्लो प्रोसेस. It can move materials across a biomembrane down the concentration gradient. Down the मतलब from higher to lower. कभी भी lower to higher नहीं होगा. It takes place in both direction. It occurs without the help of carrier protein. It does not use energy. It allows all the transferable molecules to pass through membrane, and it does not accumulate material in the cell. But in the case of active transport, it is a rapid process and it takes place only in one direction. It needs carrier protein to occur, like it, and through accumulation of material in the cell. देखो कहाँ पे होता है एक्टिव ट्रांसपोर्ट आर मूव्ड अप हिल अगेंस्ट द नॉर्मल स्पॉन्टेनियस जो हो रहा है उसके अगेंस्ट काम करें, मतलब. लेकिन अगर हम लोअर टू हायर करें तो एक्टिव पंप अगेंस्ट द कंसंट्रेशन ग्रेडियंट ट्रांसपोर्ट. Most animal cells, the protein synthesis by the ribosomes and for certain enzymatic function. So the potassium ion concentration may be 20 to 50 times greater inside the cell than outside. Still we want it. And sodium ion on the other end may be 10 times more concentrated outside the cell than inside. But both of these ionic gradients are maintained by the active transport of potassium ion into and sodium ion out of the cell. So that is, you have to know what are the difference between active transport or passive transport, that is diffusion. The next is endocytosis. Endocytosis is the ingestion of materials by cells through the plasma membrane. It is a collective term that describes three similar processes, that is phagocytosis, photocytosis, and receptor mediated endocytosis. And these processes are pathways to specifically internalize solid particles, small molecules and ions, and macromolecules respectively. All of them require energy, so they may be regarded as different form of the active transport. अब देखो फैगोसाइटोसिस में क्या होता है. फैगोसाइटोसिस का मतलब होता है सेल ईटिंग. ठीक है, डायरेक्टली सेल को ईट कैसे करते हैं फैगोसाइटोसिस. It is a common method of feeding among the protozoans like amoeba and lower like metazoa like sponges. It is also the way in which the white blood cell, हमारी बॉडी के अंदर white blood cells होती हैं जो कुछ WBCs होती हैं, हम बोलते हैं ल्यूकोसाइट, that engulf the cellular debris and uninvited microbes like viruses and bacteria. Like WBC, there are other phagocytes such as macrophages. They are found in connective tissue and liver. तो phagocytosis में क्या होता है? The area of plasma membrane that coated initially with actin myosin, they form a pocket that engulfs the solid material like bacteria. The membrane encloses a vesicle. It will call phagosome, and then detached from the cell surface, and it comes into the cytoplasm where its contents are digested by the lysosomal enzymes. You know that lysosomes are the suicidal bags. तो एक टर्म हुई आपकी फैगोसाइटोसिस. सेकंड हुआ आपका एक है आपकी एक्सोसाइटोसिस। एक्सोसाइटोसिस में क्या होता है, जैसे materials can be brought into cell by invagination and formation of a vessel. The membrane of a vesicle can fuse with the plasma membrane and extrude its content to the surrounding. Just उसका उल्टा, एक्सो का मतलब, एक्सोसाइटोसिस का मतलब बाहर जाना, एंडोसाइटोसिस का मतलब अंदर लाना, एक्सो का मतलब एग्जिट, सेल से बाहर जाना। दिस प्रोसेस कॉल्ड सेल वोमिटिंग। देखो सिंगल सेल है, अगर सेल में अंदर कोई चीज आती है उसे बोलते हैं एंडोसाइटोसिस, और सेल से अगर बाहर कोई चीज जाती है तो उसे बोलते हैं एक्सोसाइटोसिस। ठीक है, इट्स कॉल्ड सेल वोमिटिंग और एक्सोसाइटोसिस। एक्सोसाइटोसिस occur in various cells to remove undigested residue of the substances brought in by endocytosis, secrete substances such as hormones, enzymes, transport a substance completely crosses cellular barrier। जैसे किसी सेल ने अगर IgA एंटीबॉडी को अगर इट इज उसे अपने सेल से अलग करना है तो इट इज मूव अक्रॉस द सेल एंड इट इज रिलीज बाय एक्सोसाइटोसिस। कोई अनडाइजेस्टेड वेस्ट है और वो उसे अगर हमने उसे सेल से बाहर करना है, so it will form a kind of a vesicle and then it will be go out by the cell surface. So there are these exocytosis and endocytosis. So one question will come from this part: exocytosis, endocytosis, phagocytosis. Okay, so I think the rest I have already covered. One more thing about the plasmolysis, I told you that when a living plant lose water through osmosis, there is a shrinkage or contraction of the protoplasm away from the cell wall, and this phenomenon is called plasmolysis. Okay, the one question will come from this part, plasmolysis. Okay, and now sir, you please allow the students to ask the questions, because from my side I have already completed the whole chapter.
Okay, sir, you can ask them and I will just mute them if they want.